I would like to introduce David Brown. He's a postdoctoral associate in geospatial data science at the Equilab, Cornell University. His research is focused on geospatial data science methods and tools to produce location and context-specific information to support decision-making in agriculture. And today he will talk about how ranked-based data synthesis approach can be applied to Tricot data to generate new insights in crop evaluation.
Okay, so thanks again for the introduction, and thank you for inviting me to the 1000 Farms webinar series. Let's see. Okay, so I will start with a brief introduction about data synthesis of crop variety trials. So crop varieties are evaluated by or for different reasons and using different ways for evaluation, mainly for yield, and we know as reaction to pests and diseases and climate adaptation, if the varieties are drought tolerant or support or can tolerate high temperatures, things like that, but also for food quality traits. So in 2020, we published a literature review about data synthesis in crop variety evaluation. So what you could see in the left hand is the main sources of data for data synthesis in crop variety evaluation. So we have agronomic performance data mostly coming from field trials, environmental data, climate, soils, so on and so forth, but also farmer preference and consumer preferences. At the center, you could see the main elements involved in the data synthesis approach or framework, which is data management, expert knowledge, because we need a lot of domain knowledge to make sense of the results of that we get from the models, and data science. This is an updated version of this diagram. Original, we have put in instead of data science was statistical learning, which is in some way equivalent, but I think it's more closer to data science now. So then when we turn that data into information that could support the decision making of mainly three different types of users: breeders, farmers, and extension agents. So the breeders have to take decisions about which varieties to or which lines to move forward in the breeding pipeline, which traits must be prioritized, also to assess the genetic gain to select lines to move forward. As I said, in the case of farmers, they need to take the decision to which varieties are going to plant, but also which varieties are they willing to evaluate, especially if we are talking about participatory approaches. And then also take the decision if they are going to adopt a variety or not. And then in the case of extension agents, we have which varieties are going to be recommended to farmers, and then which are going through channels of marketing and distribution, regardless if we are talking about extension agents in the public sector or in the more commercial side. So why data synthesis in crop variety evaluation? Well, first of all, it's a more efficient use of the data. If we conduct study for a particular purpose, then we could reuse that data several times, and that means a higher rate of return per data point. It could provide better understanding and better predictions. As we pull different studies together, we will have a larger sample size and eventually, not necessarily, but eventually a more or higher statistical power. But also a data synthesis allows to take into account the multi-dimension nature of the data. As I mentioned before, we have several sources of data and several dimensions that we must take into account in the evaluation of varieties. So in the case of Tricot, I'm not going into too much detail here. I'm assuming most of the audience is familiar with Tricot, but we know that it's a participatory approach to test technologies supported by a digital platform named CleanUp. And then after the farmers evaluate the varieties in on their farms, and the data is collected and then uploaded to CleanUp, which is the usual approach, we have mainly two types of workflows. One is to use the statistical tools integrated in CleanUp. The other one is to use a set of tools and packages available to do a more customized analysis. And most of those tools have been developed by either members or collaborators of the 1000 Farms project. So in this case, what I'm going to present today is based on this second workflow or type of workflow that you could see at the bottom. So the first case I will talk about is the data synthesis of common bean tricot trials in Central America. So in this case, it's actually red common bean, and that's the reason why Guatemala is not included in this particular data synthesis, because most of the trials there were on black beans. But we have, for this case, we have 4 countries, 14 trials established independently between 2015 and 2018, 17 genotypes evaluated across. Not all the genotypes are evaluated in all the trials. 5 different institutions working in 4 teams. And that generated, when put all the data together, are around 1,800 trial plots, which translate into 1,800 data points. And just to highlight the fact that in this area in Central America, we have like 3 different growing seasons, and that poses a challenge for modeling environmental adaptation, because not all the trials were established subsequently or in all the across the seasons, but we have some trials in one season, other trials in another one. So the objectives of this data synthesis, the first one was to understand the main environmental conditions having an effect on genotype performance, and understanding genotype performance as the overall appreciation by farmers. So at the end of the growing season, the farmers state their overall preference about the varieties they evaluated. So which is the best and which is the worst, based in a global assessment considering several different traits. But we also aim to produce a spatial explicit predictions of genotype performance, but not only to produce those spatial explicit predictions, but to assess the uncertainty of those predictions. So just to give you an idea of the different sample size. So here on this column, you could see the different sample sizes of each trial included in this data synthesis, ranging from 14 trial plots in Costa Rica to more than 700 in Honduras. And this is to show you how those varieties or those genotypes, because we have experimental lines and release varieties, are interconnected in terms of which of them were compared directly or indirectly. So you could see some of them are never compared directly, but this is solved by rank aggregation, which I'm going to explain a bit in this right now. So this is an overall view of the rank-based data synthesis approach. So it's a combination of existing approaches and methods. So we start with Tricot data, which is basically ranking data. And we apply rank aggregation, which is basically a method to put several rankings together. We use the Plaquet-Luce model, which is a classic statistical model for ranking data, which was combined with the framework of recursive partitioning, which allows to implement the model like in a way like decision trees, and that's the example you see at the bottom. and it also allows to use environmental data to assess the influence of those variables into the performance of the variety. And I would like to again highlight here, this is possible because we have packages like Gosset, INR, and Placket Loose to fit or to use those models for this analysis. And both are being either developed by either members or collaborators of the 1000 Forms project. So we aim to characterize the environment or to assess which environmental factors or variables have an influence on crop variety performance. So we use this set of climatic indices, which are known to potentially have an influence on bean variety performance. Not necessarily are developed only for beans, but what I'm trying to say is that they are coming from previous studies that support this. And we computed this using another R package developed by um, people from the 1000 Farms, Kawe and Jakob developed this climate trends package. So it's possible to compute all those indices using that package. And these climate sensitivity indices as well are coming from literature. We adjusted the thresholds using the common bean requirements. And we computed those indices as well using the climate trends package. Finally, we also included soil variables that are coming from soil grids database. So after feeding the model, we end up with this plaquette loose tree. And the most important in this case, the most important variable was the warm spell duration index, which is the number of consecutive days with temperature higher than 19th percentile. That basically splits the main data set into two different sets of environments. And on the right hand here, you could see environments in which there are more than 4 days with a very high temperature. So varieties or genotypes performing very well here at we could say that they are like a tolerant to high temperatures. So what I'm trying to highlight here is this variety EAP 950893 has been released at the time of the study only in Honduras, not evaluated elsewhere. And the one that you see immediately below is a very well-known variety released across Central America. So it's like kind of Czech variety, if we want to consider that as well. So you could see here both perform very similar, but in a warmer environment, this one, the EAP 9508, 93, it's performing better in a more harsh environment, I would say. So when we go to the spatial explicit predictions, those are predictions at the pixel level, in this case, 11 by 11 kilometers. We see that same variety, which was released only in Honduras, seems to perform very well in Costa Rica. Now, what we see here in each pixel is the probability for each variety to be in the top 3 varieties. But we need to also assess the uncertainty. So we use normalized entropy to assess the uncertainty of those predictions. That means how certain we are about those predictions. So darker red means higher uncertainty, lighter red means lower uncertainty. So light red is good, dark red is not so good. But even when we have dark red, entropy goes from 0 to 1. So the maximum we have around here is 0.3, which is actually not so bad in terms of uncertainty. But the point I want to highlight here is that it give us an idea that, okay, potentially this variety could perform well in Costa Rica, but has never been tested there. So the suggestion there is not to go to a direct recommendation to farmers or to introduce that variety directly, but to perhaps evaluate that variety in Costa Rica. And what we have here is another measure of uncertainty, which is called the area of applicability. So basically, it's a way to assess how the model during the cross-validation process um, learns the relation between the variables from the training and then compared to the test on the cross-validation process. So it's how well the model is performing in terms of understanding the relationship between the environmental variables. So green means good, red means that those predictions that we see in the bigger map does not apply to this case, or we cannot rely on those kind of predictions for the red areas. And what we see on red here is basically the areas corresponding to protected, natural protected areas across Central America. So basically, those are areas in which actually it will be very hard, both from the agroecological conditions, but also from the practical point of view, to grow beans. So the potential application of these data synthesis and the results are targeted evaluations of top performing genotypes. As I briefly said before, instead of going from a very big list of genotypes to test, we now could have a more reduced set of varieties to plan a new evaluation in a specific location. If the predictions are in a lower uncertainty area, we could actually potentially go to direct recommendations to farmers. And this kind of data synthesis also allows to incrementally reanalyze the data and have, as I said at the beginning, have a larger sample size and the most robust predictions. So now I would like to move to the second case, which is the data synthesis of cassava tricot trials in Uganda. I will spend a bit less time on this, not because I think it's less important, but because this study was led by Anrita, Stephen, and Paula Iragabas from NACRI in Uganda. And I hope they later present this in more detail. But I would like to highlight a couple of things here. So we started with a data set of two different cycles. Each cycle of evaluations of cassava elite clones uh, was conducted in two different seasons. None of the cycles include the same set of clones or genotypes, and one check variety. So you could see that the design of the location, the experimental design of the allocation of the trials was spread across Uganda, and the aim was to cover different agroecological zones. So you could see here that the check variety is actually the one allowing to combine the two cycles and compare them. So there is no direct comparison between the varieties evaluated in the first cycle to the ones in the second cycle. So we started trying to understand which are the socio-demographic and the geographic factors that could influence the genotype performance in this case. So we started fitting the model with these three variables: gender, age, and district. So district was the most important variable there. And since location was the most important, we move on to evaluate the influence of climate. So after fitting with almost the same set of variable climatic indices I showed you for the common bean case, we end up with the number of days with rainfall higher than 20 millimeters. So that basically tell us that this panel here is showing us a set of environments that we could consider weather environments. And this particular variety is performing well in a kind of less, or I won't say dry, but relatively drier environment compared to um, how it performs in a wet environment. But still in the wet environment, it doesn't perform very differently to the Czech variety. But in this case, the aim of the study was also to evaluate food quality traits. So samples from the varieties evaluated in the field trials on Tricot were evaluated also using Tricot with food products made in the traditional way that cassava is prepared in Uganda, and then labeled in this way to us so the farmers could assess the same way as a Tricot trial. So to have a comprehensive evaluation of both agronomic and food quality traits, we used this large heat map, and what you see in the numbers are computations of reliability, and reliability is the probability for a variety to outperform the Czech variety. So here we have the Czech variety in the middle, cycle 1 in the upper panel, and cycle 2 varieties in the lower panel. And basically, numbers or cells in more bluish background means higher reliability. More reddish colors means lower reliability. And numbers in bold means that that specific variety is statistically different to the Czech variety in terms of the reliability for that particular trait. So we could see that some of the varieties perform better for agronomic traits, measured mostly in the early stages and then 12 months after planting, we have a bit more food quality traits. But this allows eventually the breeders to evaluate which variety is performing better in a more comprehensive way. So the potential applications of this is to, as I said, top performing varieties could be identified for both agronomic and food quality traits. And then the release of the varieties is based on data. So I would like to list these two publications that support the two cases that I presented, not only to invite you to read the papers, but also to acknowledge all the people involved there. It will be very difficult to list all the people that contributed working in those two cases. And I really appreciate, in the first case, I was leading that study. In the second case, I was a collaborator. So in the second case, I really appreciate them, the leading authors, to allow me to participate. And with that, I will thank you and will be happy to answer some questions.
Thank you, David. Very interesting presentation, very clear. And so now we can move on to question session. If you would like to ask David a question, please raise your virtual hand. I have one I can start off with, David. I was just wondering how feasible it might be to incorporate future data, for example, on the common bean study in Central America, if you then came across some common bean, red bean data from Africa, would you be able to incorporate that? Would it be meaningful in any way?
Um, Thanks, Rachel. That's a very good question. In terms of feasible, as long as we have some linking, as we saw in the case of cassava, actually there is the check is the link between the two sets of varieties. So as long as we have some variety that we are pretty sure it's has been evaluated in both regions or locations, um, that will allow us to eventually run the model, I would say, um, with some constraints still, but that will be in a way feasible. But the most interesting part of your question is how meaningful would be. That's a hard question that I'm like actually not there to answer right now. I mean, it will depends on a lot of factors. I will say that how related are genetically the varieties in some way, and so you could identify some link there. But I don't know. It will be interesting to see what happened.
Thanks very
I th
much.
Yeah.
Moitala, you have raised your hands. Please unmute and ask your question. Moi, would you like to ask a question? You can just unmute yourself, please. Okay. Maybe he's having a technical problem. Well, in the meantime, any other questions, comments for David? Edward Carrier said, no questions. Very wonderful presentation.
Yeah, maybe just a
Ah,
brief one. This
yes,
is Williams.
go ahead.
I, I'm trying to reflect on how to extend this to industry kind of based uh, testing. If we are breeding, say, for industrial products, and you want your feedback much more from people who are processing your varieties than from farmers. How will it play using Tricot?
And thank you, Williams, for the question. It's very interesting question indeed. I will say first of all that yeah, well, Kawe is already helping me with the answer. But that was part of my disclaimer. I'm usually involved mostly in the data science and the analysis of the trials, which what I'm trying to say is that I'm not usually establishing the trials or planning the trial itself. But what Kawe is saying is that they have done some processing trials in Nigeria with cassava. So I'm assuming it's possible, and they have taken into account or they have took into account the requirements for processing. So yeah, in the sense from the data science or data analysis part, I will just add that tailoring the information that we get from the analysis to that different type of end user, which is not a farmer, but the processing um, or the processors, I would say that perhaps will be a different framing of the results.
Okay. Kawe, do you want to complement that or say anything about your experience with cassava in Nigeria?
Yeah, just to say that congratulations, David, on this. So just to say that what he presented here is more from the, let's say, crop ecology point of view, But from the consumer point of view, like thinking about the application of the product and also market orientation, this is also possible. We have some literature on that, so feel free to reach out. So then I can send most of the work that we've been doing, and mostly the work is in cassava and yeah, most of the work has been in cassava for this type of studies.
Okay, thank you, Kawe.
Yes.
Any other questions? Okay, I don't see any. So if there are no further questions, we can wrap this up. And let me just close by once more sharing with you the links for our Thousand Farms project resources, our website, social media, and the community website is the last one where you can find all of the recorded webinars and the question and answers and any relevant links. So what we can do also is put a link in there to the papers that David referred to at the end of his presentation. So for some further reading. So with that, I will say thank you very much, everybody, for joining us today. And we'll see you again in 2 weeks for our next webinar. And thank you very much to David as well for presenting.